All right. Good afternoon, everybody. I'm pleased to introduce Toni Jordan-Millett, who is an intern in the plant ecology program. So Toni graduated from high school in Maryland, and she graduated from Rice University in 2022 with a BS in cell bio and genetics. Her numerous experience prior to coming to Archbold include working in the desert at the Savieta LTR site on grass endophyte interactions with her advisor, Dr. Tom Miller. Toni has been a wonderful member of the plant ecology program, who is known to us in the plant lab for her toughness in the field and her positive attitude. She holds what we believe to be the world record for counting Dicerandra fanicula seedlings in a single plot at over 1,400 is the last count. And she also has a notably deep love of Tootsie Roll Pops, which keep her charged in the field. For her intern project, Toni wanted to delve into plant microbe interactions in the scrub, which are incredibly important for shaping the scrub system that we know. Among many other things she accomplished and will share with you today, Toni did quite a bit of experimentation with lab protocols for staining mycorrhizae, and these are going to be incredibly helpful for us going forward, in addition to all the work that she's showing today. Finally, I'm pleased to share that Toni will be staying somewhat nearby and starting a technician position at the Neon site in Gainesville this summer. And with that, I'll hand it over to Toni.
Hi, everyone. So thank you for that introduction, Erin. Let me just close this. So good afternoon, everybody. So today, as you can see, I'm going to be talking to you about our scrub plants and specifically the mutualists that help them live. For those of you who don't know much about Florida scrub, it's one of the habitats the major habitat we have here at Archbold, and it's a threatened ecosystem out here in Florida. So the title of my talk is Tough Scrub Plants and the Fragile Fungi That Help Them Thrive. So out here in the scrub habitat and other high stress habitats, there are a variety of environmental stressors that plants have to deal with. So even as they're eaten, trampled underfoot, exposed to high temperatures, plants have the extraordinary ability to live and thrive. So my major question is, how are plants capable of persisting in stressful habitats? Habitats like the Florida scrub. And so what we happen is across the Florida scrub, there are three major white sand scrub habitats. There are the flatwoods, scrubby flatwoods, and rosemary scrub. So what happens across these habitats is that elevation is variable. And so what happens is it's at its lowest in the flatwoods. And as we move across the habitats, it increases, being at its highest in rosemary scrub. And so while all plants within these three habitats experience a combination of different kinds of abiotic stressors like low precipitation, well-drained soils, high temperatures, several of these stressors can actually vary with elevation. So what we're seeing is that as you go up in elevation, you tend to have decreases in vegetation density, nutrients, and water availability, which means there's an increase in stress levels. Our highest stress habitat being rosemary scrub. And something important to note is even within a single habitat, these elevational differences can lead to changes in stress levels. So back to our major question. So how are plants persisting in these stressful environments like the Florida scrub? So plants will use a variety of things we call adaptive life strategies. So one strategy plants use is mutualism. So this is basically when a plant's exchanging resources or giving resources to some sort of organism, and in return, they get some benefit from the organism. So there are different kinds of mutualists that are out in the environment. Things like you're probably thinking of, like pollinators or insects that are going to protect against herbivory. And then there's also microorganisms that are present within the plants, in the leaves and the stems and the roots, things like endophytes that help protect plants against different stressors. And then we have our microorganisms and microbes that are present in the soil that will help plants get the nutrients they need, things like mycorrhizae, for example. So I wanted to look at, is mutualism an adaptive strategy? And to know if it's being used as a strategy out here in these habitats, we can examine its relationship to stress. So if a plant needs mutualisms or needs mutualists to live in a higher stress habitat, that as stress increases, plants are probably going to be focusing on mutualism. But sometimes this doesn't happen. As stress increases, the mutualists are also becoming more stressed and plants aren't able to invest into them. So mutualism begins to decrease or remains unchanged. So plants then have to engage in other kinds of strategies. So for my research questions, I wanted to look at specifically, is stress correlated to mutualism? Yes or no? And then what kind of stressors are actually interrupting this relationship between stress and mutualism? Things like the allelopathic rosemary that's out in the rosemary scrub habitat, releasing chemicals into the ground that interfere with this relationship, or things like our roadside habitats, our fire lanes that are highly disturbed and are also possibly altering this relationship between stress and mutualism. Additionally, I also wanted to look at, does this relationship between stress and mutualism differ between a generalist species that's present across many kinds of habitats and a specialist that's only present within a single habitat? So for my study system, I looked at specifically fungal mutualism, specifically mycorrhizae or endomycorrhizae, otherwise called AMF, which is located below ground. So what happens is these guys are below ground, they come in contact with a plant, and then they'll start colonizing the plant, forming these structures inside the plants, and specifically inside the plant roots. So for my study system, I used two species. I had my generalist species, Baldwina angustifolia. It forms these really pretty yellow flowers, otherwise called coastal plain honeycomb head. And then I had my specialist species, or Hypericum cumulicula, or Highland scrub St. John's wort. Very long names. So as you can see, looking at these green specks across Florida, the generalist species is located across many places in Florida and across many different kinds of habitats, while the specialist is located in a specific number of counties and within a single or few number of habitats. So for site selections, this is a map of Archbold that you're seeing, or the research station that we're at, and I went ahead and sampled across rosemary scrub habitat, or rosemary balds, and I tried to get an even distribution of sites marked in red that I'm soon gonna highlight a little further, spread across the station, and I wanted to get an even distribution of different kinds of elevation or really stress across the habitats. So I used two roadside habitats, or two roadside balds, to look at the effect of disturbance on this relationship between stress and mutualism. And then I also selected 8 more in balds that aren't roadside to further examine how rosemary is going to affect this relationship between stress and mutualism. So for sample selection, you go out to these sites and you see different kinds of plants. I look and spot the plants that I'm gonna wanna pick, and I'm only gonna choose plants that aren't a part of any monitoring sites. So all the endangered species, sometimes we have flags out there that are marking which ones we study and monitor throughout the year. And then I'm gonna try to get an even distribution of 2 different age classes, both adults and juveniles. And then I also looked at different kinds of distance to rosemary, or different kinds of experiences with this allelopathic chemicals in the soil. So they were either going to be really close to rosemary or far away. And then I also looked at, as mentioned before, being in bold or roadside, different kinds of disturbance. So for sample collection and processing, I see the plants that I want. I go out here and I carefully dig them up and out of the soil. I bring them back to the lab to image and take pictures of for processing. And then I start the processing. So I go ahead and prep these roots for a stain where we can finally see the fungal mutualist, or AMF, inside the roots. So what happens is I'm able to soak them in KOH. That just clears out everything from inside the roots, all that gunk. And then I can stain them using a vinegar dye solution to look at AMF and see these really cool structures. So what happens when an AMF starts colonizing the plant, they form different kinds of structures, things like spores that are these spherical structures on the outside of the root. that we're able to see looking at underneath a microscope once the roots are stained. We're also going to see things like hyphae, that are these thin, wispy kind of lines that go in between the roots and inside the roots. And then we're also going to see things like vesicles that are really inside the root, that are these smaller circular spherical structures. So what I want to do then is I'm seeing this AMF, I'm seeing this colonization happening, is I have to get a quantitative, association so I can do some form of analysis. So what happens is I take all these small segments of roots that I've cut up, stained, and we get them onto a grid. And so once they're on a grid, we're able to look across the grid and see, is there AMF, how much AMF we're seeing, and basically assign a score. And so what we're doing is we look at these intersections with the grid. Is there AMF? Is there no AMF? How many root intersections are happening? So for example, if the dark blue is going to be AMF for the mutualist, then we can see that at the intersections there are 5. And if we're looking at the total number of root intersections, it's going to be 27. We can multiply that by 100, and we get this percent colonization, or this value we can use to analyze. So after performing all my analysis, I was able to see that there actually isn't a relationship between stress and mutualism. So what we're seeing here is that as elevation increases in these rosemary scrub habitats, percent colonization by mutualists is pretty much remaining the same. There's not a lot of changes for both species. There's the Baldwina, or the generalist species, in red, and our Hypericum, or our specialist species, in blue. So what's happening when we look at, okay, what are these stressors like rosemary going to do to mutualism? Is it changing this relationship between stress and mutualism? So as we see, we would think that rosemary or allelopathy as a stressor would disrupt mutualism, would decrease it for both species. But what happens is that as you get closer to rosemary, it appears that mutualism decreases for both species, but really it's only statistically significant for our generalist Baldwina. So this actually makes sense because a generalist species isn't found only in rosemary scrub habitat. It's actually found in a variety of habitats instead of our specialist, which is only going to be or primarily found in rosemary scrub habitat. So its relationship with the mutualist isn't going to be as affected by allelopathy. This makes sense because that's where it's commonly going to be. So then we go ahead and look at our roadside plots, or our disturbance. How is that affecting stress and mutualism? And so while it's not statistically significant, it seems like there's this trend that roadside plots or disturbance are actually increasing mutualism for specialists. So this kind of makes sense given that a lot of our rare endemic species or specialist species grow in rosemary scrub and roadside plots for some reason. These highlighted to disturb fire lanes that a lot of other plants aren't growing in. So mutualism here is super beneficial in this area for specialists, or for a specialist, we should say. And then in general, as a general result, outside of my specific research questions, I went ahead and looked at, okay, we have percent colonization or mutualism. Is it really benefiting the plant? Is it increasing the success of the plant? And so how we do that is we're going to look at reproductive structures. So the number of flowers and fruits, and these are going to be signs of success in a plant. So is it increasing for either species? Well, we can see that as percent colonization increases, there's an increase in the number of reproductive structures for Baldwina, our generalist species. And we would think that mutualism would help any kind of plant species to be more successful, but we're not seeing that happen. So this is something that opens up more questions. So in summary, is stress correlated to mutualism? What's happening there? Well, no. Fungal mutualism is likely not correlated to relative elevation in rosemary scrub habitat, but do keep in mind that is a single kind of habitat that we have out in the scrub. We have other kinds of habitats. And then what stressors are affecting this relationship between stress and mutualism, and just altering the levels of mutualism within plants. So allelopathy and disturbance are both affecting mutualism within plant species. And then I wanted to look at, does this relationship differ between a generalist species and a specialist? And possibly in the face of different stressors, such as allelopathy, we're actually seeing a greater effect on mutualism in our generalists, which aren't as well adapted to and specialized to the rosemary scrub habitat. And then we're also seeing that there's an increase in mutualism within our specialist species that are found in our roadside habitats. So in conclusion, the colonization of plants is broadly consistent across rosemary scrub habitat. And Florida scrub endemic species are possibly being better adapted to certain stressors such as disturbance and allelopathy, which makes sense because that's where they're located. Even though my results suggest mutualism might be the only factor behind their presence of these specialist and scrub endemic species in rosemary scrub, I still think that given the little above ground overlap in these habitats, below ground interactions are important. Other below ground adaptive life strategies should be studied, and fungal mutualists and other mutualists below ground should be something that we should consider. So finally, for my acknowledgements, I want to thank Dr. Aaron David for being a great mentor here and a great advisor for bouncing around all sorts of ideas while I was here. And then I want to thank Shiram for helping me with R and GIS. And I would like to also thank Dr. Mark Dayrup and Nancy for allowing me access to a lot of their imaging that I was able to take in their lab. I often kicked them off the computer at really bad times, and they were really nice about it. I would also like to thank Hannah, Sterling, Andy, Layla, Jeffrey, and all the previous Plant Lab members, Christine, Iris, for just making my time here really great. I also want to thank all our residential technical staff, Kevin, who I don't think is here today. We got to do some really cool stuff with prescribed fires, which you can kind of see. That was great. And then he also does a lot of work for us. Bert, Lupe, Verna. I want to thank Viv and Julie. Without their work, well, I used their maps to get to all my sites, and that was extremely helpful, especially during wet season, so I didn't get stuck. I want to thank Sharon, Nova, and Laura. George, who isn't here today, but he helped take so many pictures and bounce around ideas. I also want to thank Dr. Hillary for also, of course, being a great support to everything I do here. Archbold for their funding and support. And I want to thank all my friends and family who helped me get here and who are present today watching my talk. With that, that's it. I just want to say thank you everybody for coming, and I'd like to ask if you guys have any questions. Okay, let me turn this on. There we go. There you go.
Questions?
I was like, does anybody have any questions?
So what was the hardest part about this project?
Okay, yeah, thank you for asking. So originally when I was first designing this project, I really wanted to look across a wide variety of habitats, specifically our oak species, because they're present in pretty much every habitat. But it turns out trying to clear out these really dense, thick roots is really hard. So I had to basically just experiment for the first 2 months I was here. I was just experimenting over and over, trial and error. What species can I work with? What concentration of chemicals do I need to use? For how long? Do I need the heat? It was kind of tricky figuring all that out, and it took a lot of time.
Kind of two questions. Seeing that the specialist species didn't seem to benefit reproductively from the mutualism, do you think that there's other benefits it could have gained? Maybe like something to do with the plant growth, like from the leaves or the stems and stuff?
Yeah, that's a great question. So I was thinking about this too. It's really strange that this plant isn't getting this really great benefit from mutualists. And so for Hypericum specifically, the height of the plant doesn't really significantly differ amongst all the Hypericum that we have or the individuals that we have. But maybe there is some sort of benefit that I'm just not seeing. maybe in its root length, which I'm going to be studying at some point later on, but I have to analyze all the images, or something else, maybe something that's happening below ground that it's doing that's really beneficial that I just don't know yet.
I think you also looked at adults versus.
Yeah, I also was looking at to see if there's a difference over time. Like, is mutualism still happening when these plants are just a couple months old? And it turns out that pretty much, yeah, these mutualists come in, they start interacting with these plants really quickly, even when they're really young. And yeah, these mutualists are still important. Even though there's not much of a change, they're still there, and they are just present across all the habitats, helping these plants survive.
This one over here. You already had a turn.
What's your favorite plant and why?
I knew this question was coming. I'm so excited. So first off, for everybody that's online watching this, so here where I work, there's just a whole bunch of threatened species that not a lot of people get to see in their lives. And it's really cool being able to work here and just go out and, oh my gosh, there's a pine lily. Oh my gosh, there's Dicerandra, all these species that I just wouldn't see anywhere else. But the very first flowering plant I saw that was a rare species was actually scrub morning glory. And I remember looking at it, and it was blooming at the time, and it's a really pretty flower. I was just like, oh my God, that's so cool. And I was just like, Auntie, what's the name of that plant? And she told me, and I forgot it. But then I had it down like 2 weeks later, and I was like, okay, I like that plant. That's probably one of my favorite plants, but I definitely have more favorites. I mean, we go out here, we work with plants for like hours. So you have to like plants to do that. So there are a lot of plants I just love.
So at Archbold, there's a variety of generalist plant species and specialist species. Why did you pick the two species that you did?
Yeah, so I didn't mention this, but there are some previous studies involving looking at microbe interactions for Hypericum, things like changes in the nutrient level in the soil that we haven't done for other species. So I thought that this would be a great thing, or a great plant to look at, especially. And then for my generalist species, I kind of went out and I was out walking. And at the time when I was first thinking, okay, I need a generalist species, I kept seeing Baldwina flowers like everywhere and just randomly across the scrub and the rosemary scrub. And I was like, okay, we're working with that. And so the super cool thing is that Baldwina actually is one of these plants that only lives for 2 years. And so even then, there's still these really cool relationships happening between stress and mutualism. And I can't imagine how different or how crazy it'd be if I looked at a generalist that was more long-lived, like the oaks. That would be something that would be really cool to do in the future. Yeah.
Great talk. Why do you think it is that the mutualisms is increased in the road disturbed habitats?
Yeah, so that was a super cool result. It's just, although it is just a trend, and that is kind of crazy. I actually don't know why, because you would think that, okay, we have this sand and these cars are coming over, driving things over, disturbing the sand, moving it. You would think that these things underground, these networks are actually going to be disrupted or broken apart. But it's weird that for some reason, maybe the fungal mutualists that are associating with these plants are actually, maybe it's a certain kind that's like, this is AMF is just a broad category, but there's different, species that are out there. Maybe there's a certain kind that's actually a lot sturdier or something. I don't know what's happening, but that's just an avenue for more questions.
All right. I'll read a few
All
from
right.
Zoom. Let's see. Is there any biological reason you might speculate that percent mutualist colonization would decrease in the specialist species on that last figure? I think that's the reproduction one. Probably.
Why? Okay, I don't know how to go back to pull up that figure.
Sometimes the arrows.
Okay, we're gonna manually go back for the sake of everybody here. Give me a second. Okay, so we're talking about this figure. So reasons why the percent colonization is, this is kind of the question that was asked earlier about why are we seeing this decrease in percent colonization and really just less reproductive structures. And I'm not entirely sure, but it's possible that at some point in time, when these plants are growing and aging, at some point in time, maybe if I'm assuming that there's more success as the plants age from year to year, that maybe there's this curve happening where the benefit of mutualism on reproductive structures is actually going to start flatlining or maybe even falling. That's just my theory, though. I don't know what's happening specifically. I would have to study it more.
All right, and then we got one from Eric Mangus. Nice talk, Tony. I wonder how you think your study could be expanded to more than 2 species and to more habitats. Can a
Oh.
broader study be practically done given all the work involved?
Yeah, so I have thought about this many times. I think it would be fantastic. It's just for below ground studies, honestly, there's not a lot of studies that have been done into, this is kind of a new field of just looking at the microbes, looking at the mutualists that are below ground. And that's partially because my study is a little invasive. We're pulling up these plants and endangered ones and looking at them underneath the microscope. And for me, a round of processing plants and looking at them and scoring them takes about like a week or even a week and a half. And that's just for, let's say, maybe 20 plants. It's just pretty time intensive trying to analyze these plants. It just takes a lot of time. But if someone was able to come in and look at more habitats and see, oh, we're in the super high stress habitat, but what's happening across many habitats when there's more of a stress gradient, what's happening to the mutualists and what's happening between mutualism and stress. And I'm sure that if I was able to, it would have been great to look at more species to have just to see what was happening. Is this something we can generalize to more generalists, more specialists? But
And
yeah,
then
it'd be great.
we got one more from Tom Miller, who's tuned in.
Oh my gosh, hi Tom.
He says, great talk, Tony. Do you think the variation in fungal colonization, e.g. near versus far from rosemary, reflects variation in availability of fungi in the soil, or is it more about the plant investing or not in fungal partners?
So that's a super good question. This was something I kind of wanted to look at. So originally I had actually collected soil samples to see the level and variety of AMF and fungal mutualists that are existing in the soil, not even associating with plants, just existing, to see if that was happening. But I wasn't able to finish that analysis. So all I have right now is this data between this relationship. And honestly, I think it could go either way. It could be that allelopathy is making it so that these spores can't live out there. These mutualists aren't able to live out there. Or it could be that they're maybe these mutualists are super, super hardy. They just exist. They're able to tolerate these super low nutrient conditions or this allelopathy, and it's only affecting the relationship between the plants. Because no one's really dug up a whole bunch of spores and looked at what's happening out here in the scrub with spores. I can't say for sure what my thought would be, but I'd probably lean towards it's affecting more so the relationship between the plants and the mutualists. I would assume that the mutualists themselves that exist in the soil, the microbes and microorganisms, have a little bit of an easier time, but I'm sure they're still stressed to some degree by allelopathy.
I'd also add that I don't think we really know what the rosemary itself associates with. That's a
Yeah,
big
no one
knowledge gap.
knows. I'm telling you, if anyone wants to come here and do some studies, please do, because there's so many things that we just don't know yet. Yeah.
One more?
I liked the stained root picture that you had labeled.
Oh, yes, yes.
It wasn't what I expected mycorrhizae fungi to look like. I thought they would be strings. These seem like independent and they're also different shapes.
Mhm.
Could you just explain that a little bit? Are they all different species making different shapes? How are they connected?
So it could be different species, but generally, when any AMF comes in contact with the plant, first it's gonna be the spores. These are just, they exist in the soil. They come in contact with the plant and they start forming these links. Now, usually what you're gonna see is, you're right, it'd be more interconnected, but by these hyphae that you're seeing, the strings. But what happened when I was, staining these plants, my process is like really just, it's a really intense and stressful thing to do to the roots. And so there's probably some breakage happening. The stuff's still there, but the connectedness, they're supposed to be connected within the root, but it's just getting broken apart a little bit by the kind of stains I do. But Erin might also know more about that's pretty much what's happening. But yeah, these it goes inside, this is why it's called colonization, because it comes in and it invades these roots, it goes in and it makes these other structures inside the plant and across the plant. Both on the surface and inside. Yeah.
How to ID those species, which is to say they're very hard to ID.
Oh, yes, I tried doing that. I wanted to look at all the kinds of species that we have, or maybe if there was a variety. And when you collect spores and you have to do some centrifugation and different things to basically separate them from these guys are like tiny. You can see the roots and they're bigger, but this is a millimeter. One little square is a millimeter. So these things are extremely, extremely small. And when you try to separate out everything from the soil and look at them and see, okay, what species are we having? And they're not stained, you're seeing just blobs. And it's a really good skill. And it's something you have to be really experienced with to be able to ID and say, hey, there's this species, there's that species. And knowing how much variety and biodiversity we have here, it's probably likely that there are some specific species of AMF that are present just here. And I think it would take more than a lot of time to really get down to, okay, these are the species that are present here. And these are the species that are not just present in Florida, but specifically in the scrub.
Right. One more question? Anyone has one? Well, I think Christine had one question.
Okay.
What's your favorite memory? So we can conclude on that one.
My favorite memory, like at Archbold, or I guess
She said in the scrub.
in the scrub. Oh
Specifically.
gosh. All right, what happens in the scrub stays in the scrub. Many things happen, but my absolute favorite thing, oh gosh, I would have to say there was a day that say, Sterling knows what I'm talking about, and Christine knows this story actually, where we were wandering the scrub searching. We were searching for a specific plot, and it was you guys probably don't know this species. It's one of the endangered species we have here, basically. We were looking for a specific plot, and we were wandering for hours, and we're trying to figure out, where is this? This happens when you're out in the scrub. It's hot, you start going, you're like, where is this thing I'm looking for? And when you finally find it, it's like this aha, just hallelujah moment. And that was both a hard day, but also just amazing. I remember being like, oh my gosh, we finally found it. There they are. And then I think the pot was empty or something, but that's okay. It was there and we got it. We were able to go home. It was fantastic.
That's a good note to end on.
Yeah.